Remember? Right? Okay. So we left off talking about the cardiac conduction system. We left off talking about how does the heart get a signal to contract, okay, so that it can beat and circulate blood in the proper way, right? I do want you guys to know the location of the four components of the cardiovascular conduction system, and I do want you to know how the signal moves from one to the other to the next, okay? So SA node to AV node, down the AV bundle, and then it spins and reverses back up through the Purkinje fiber. Having it this way allows us to spread action potentials quickly across the heart, and because of the anatomy of the heart and the way the action potentials are going to be transmitted, we're going to get this very distinct, because it starts in the atria. The atria will contract first, and then the ventricles will contract second. If that doesn't work right, which happens in some cardiovascular diseases, or when you have some kind of what we call arrhythmias, where the conduction system isn't going correctly, if all of these four chambers contracted simultaneously, one wouldn't go anywhere, right? The atria have to contract to move blood into the ventricles. And then they have to be relaxed so that they can refill. And then the ventricles have to contract to get the blood to go out. And so basically, if they're all going at the same time, blood can't go anywhere, right? Because it's all going to move via pressure. So that becomes really, really important. And I'll show you guys the EKG in a minute. But in some kinds of cardiovascular disease, when you have certain kinds of arrhythmias, that normal SA to AV bundle to Purkinje fiber, that normal kind of order of all of that can get messed up. And then the heart can't pump blood correctly, and that's when you have a heart attack, right? That's when you're going to have a heart attack. Okay, so one heartbeat, we call one heartbeat physiologically a cardiac cycle, okay? Cardiac cycle. So in a cardiac cycle, you are going to have these individual periods that each chamber of the heart will experience, okay? So during one cardiac cycle, all four chambers will experience a period of what we call systole, and all four will experience a period called diastole. Systole stands for or denotes contraction, okay? That's when that chamber is contracting. Diastole denotes relaxation, okay? So blood moves all through the body via differences in pressure. And so when a chamber of the heart contracts, it raises the pressure in that chamber. And that's going to raise pressure there, and it's going to drive blood from that area of high pressure to an adjacent area where the pressure is lower, okay? And so what happens then is that we're going to typically get atrial systole, so the atria contract, while the ventricles are experiencing diastole. And then vice versa. While the ventricles are experiencing systole, the atria are experiencing diastole, okay? So in a normal cardiac cycle at rest, so you guys sitting there right now, we spend about 60% of the time of a cardiac cycle with your chambers relaxed, okay? So it's about a 60-40 contraction to relaxation. So if you imagine that you have a resting heart rate of about 75 beats per minute, that means that a cycle takes about 0.8 of a second to occur. And so 60% of that 0.8 of a second is relaxation, and 0.4 of that 0.8, or 40% of that 0.8 is going to be contraction. Okay? This is important. And I'll try to explain to you guys why this is so important. As heart rate increases, okay? As heart rate increases, if we ask Holly to stand up and do jumping jacks for us, which we won't, if we ask her to stand up and do jumping jacks for us, her heart rate would go up. And so now it wouldn't be 75 beats per minute, it might be 125 beats per minute. As heart rate goes up, the time that it takes to perform one cardiac cycle gets smaller and smaller, okay? And so because that time gets smaller, then the amount of the physical amount of time spent in systole and diastole is going to shrink. And so all of you have a maximal heart rate. All of you have, and it's usually related to your age, okay? Most of you have a max heart rate of somewhere around 200 beats per minute. So you take 220, you subtract your age from it, that's your age predicted max heart rate, right? So as you near your max heart rate, which very often happens during a high intensity exercise, okay, one of the things that happens is that diastole becomes so short that we can have limits on the amount of blood that can get into the atria and then get into the ventricles and fill them up, because that period of relaxation is so short. And that may put a limit on kind of what our cardiac output can be at the high end, which I'll explain why that's important in a few slides. Okay? So understanding the interaction between sort of these periods of systole and diastole and how long it takes a cardiac cycle to occur based on your heart rate has some really important implications for exercise. Okay? Okay, so I'm going to show you guys this, and we'll walk back and talk about this. You guys have seen an EKG before, is that right? I assume you guys see these in physiology class probably, or maybe you've seen them in personal training or something, right? Okay, so an ECG, or it's also sometimes called an EKG, stands for an electrocardiogram. And what this is we put a series of electrodes on your chest and your back that sort of surround the heart with these things. And we can measure the action potentials moving across the different chambers of the heart. Okay? There is a normal pattern to this. If you have normal cardiac conduction system function, your EKG should look a certain way. Okay? If you have different kinds of cardiovascular disease, if you have problems with the SA node or the AV node or something with conduction, they will show up by making your EKG look different than what this normal one is going to. All right? I'm going to walk you guys through what each of these things denotes, but that's all that I want you to know. That's all I want you to know. We're not going to talk about arrhythmias. We're not going to talk about what if, say, this segment between S and T gets prolonged, or if it gets shortened, you have a sort of double P wave. We're not going to talk about what any of those arrhythmias actually mean, or kind of what's wrong with you if you have one. I just want you guys to appreciate the fact that we use EKGs to help determine cardiovascular function, especially in people that are either at risk of having a heart attack or that have just had a heart attack. All right? If you just had a heart attack, or they think you just had a heart attack, they're going to get you stable at the hospital, and then they're going to put an EKG on and make you get on a treadmill and exercise to see if they can make you basically have another heart attack, right? So they can see, can we see some sort of change in your EKG that would tell us where in the heart are you experiencing a lack of blood flow? Where in the heart might there be conduction failure? It helps us diagnose what is wrong, okay? Even though to do that, we almost have to make you have another heart attack, okay? So when you hear people talk about doing a stress test, it involves this plus exercise, okay? Put these things on, get your ass on the treadmill, let's go, okay? We don't care that you were nearly dead 2 hours ago. You're stable, we're going to do this. Okay, so on an EKG, all right, what we're going to see are these three different kind of primary waves or complexes: a P wave, the QRS complex, and the T wave. So it looks like this, okay? at the beginning of a cardiac cycle, we are going to have the SA node fires that depolarizes the right atrium. That is going to be most of the signal that is picked up here in the P wave, okay? So electrical signal, this is atrial depolarization. Then you get this weird looking thing, which is called the QRS complex, okay? What we are seeing here is going to be the signals that lead to depolarization of the ventricles, okay? So transmission through the AV node, down the AV bundle through the Purkinje fibers. That's QRS, okay? Typically, R is the thing that denotes ventricular depolarization or ventricular contraction, R, okay? Then we have a T wave afterwards, which is going to be the repolarization of the ventricles, okay? Sometimes you'll see these things go in a different direction, like you'll have a T wave, but it goes down. Part of that is which electrodes are you looking at and the directionality of movement of the signal between the different electrodes. Not that big of a deal, okay? So, for instance, let's just say you had trouble sending signals through the AV node, you would get a P wave and then maybe not a QRS, okay? Or you have ischemia in the ventricles, which is very, very common, and you get this weird thing that happens between S and T, where it sort of stays down here, and then it eventually comes back up. It's called S and T segment depression. That's one of the sure signs of that you have ischemia from blockage of coronary arteries and those kinds of things. So we could talk a whole bunch about all of that. When I taught chronic disease in previous years, in that class, we would go through different arrhythmias. We'd look at different tapes and talk about kind of what all those things mean. You guys take chronic disease now that they've taken it out. Probably best to take it out. If you take exercise testing and prescription, you guys will do EKGs. You will get an EKG performed on you, okay, while the person is exercising. You want to work in cardiac rehabilitation, or you want to be an exercise physiologist that works in a hospital, you do a lot of stuff with EKGs and a lot of stuff with exercise testing, okay? So there are master's programs that basically involve only learning how to do these things and diagnose stuff off of all of this. Okay, we talked about that, we talked about that. Okay, so we have a cardiac cycle. Cardiac cycle denotes heart rate, okay? One heartbeat. The number of beats you get in a minute, that's what we're going to call heart. There are some other cardiovascular variables that are really important for determining function and determining kind of how well you're going to be able to exercise. One of those is something called stroke volume, okay? Stroke volume represents the amount of blood that is ejected or that leaves the left ventricle every time it beats, okay? So this is blood leaving the heart during each heartbeat, okay? You guys are probably too young to have ever seen these things. So my analogy is that I got to find a movie. How many of you have ever taken a picture with a real camera that has real film in it, like old school film, okay? Many of you have. Okay, excellent. Do any of you remember the little canister that film came in, or that you had to put the film, pull it out of the camera, and put it back in to get developed? So these little black plastic cans, and they have a gray top on them usually, okay? It's about yay big and about yay around, all right? So I have many questions now about how you guys have used your cameras. I'm fascinated. Usually I get like one person in class that's like, I've heard of a camera that takes 35 millimeter film in those ways. When my wife and I were in college, there was a photography class. You could take class, you had to take pictures, and you had to go and actually develop the pictures yourself, like in a dark room, and those kinds of things. So it wasn't just, the pictures were worse than whatever your phone was. So in an average person at rest, stroke volume is about 80 milliliters. Sorry, I was buried in the weeds. That's about as much fluid as would fit in one of those little 35 millimeter camera film cases. Okay, so it's about 80 milliliters. I of course don't have anything that's even remotely close to kind of whatever that's going to be. So that's how much blood leaves your heart every time it beats. Okay, give or take. If you're a little bit smaller of a person, it may be a little bit less. If you're a larger person, it may be a little bit bigger. Stroke volume is dependent upon the size of the left ventricle. If you have a bigger left ventricle that has a larger space in it, then you can fill it up with more blood. And therefore, it can eject more blood every time it beats. Okay? One of the largest and most important adaptations to exercise training is a change in stroke volume. Left ventricle hypertrophies, the space inside of it gets bigger, it gets stronger, and so people that are highly aerobically trained have much larger stroke volumes than people that are not as trained. Okay? You can calculate stroke volume in what I always think is kind of a strange way, but it is the difference between what we call end diastolic volume and end systolic volume. End diastolic volume is the volume of blood that has filled into the left ventricle during ventricular diastole. So while the ventricle is relaxed, the atria contracts and it pushes blood into the ventricle. Right before the ventricles contract, however much blood is in there, that's end diastolic volume. Okay? So imagine these two things, okay? I have these two. In diastolic volume, this is my left ventricle, okay? This is my left atria. I'm pouring atrial contraction. I'm pouring blood into the ventricle. It's going to fill up to some extent, right? Then the atria relaxes, so it stops. And then now the ventricle is going to contract, and it's going to dump the blood out. So however much is in the ventricle at the end of atrial contraction, that's in diastolic volume. In systolic volume is the amount of blood left in the ventricle when it stops contracting. So I've got my blood in the ventricle, I'm going to contract it, so I'm going to squeeze, and it's going to shoot a certain amount out. Under resting conditions, kind of non-maximal or kind of sort of heavy exercise conditions, there's always going to be some blood left in the ventricle when it finishes contracting. So if you take the difference between these two, that tells us how much blood left. Okay. Ejection fraction, which is a really important measure of cardiovascular function, is the percentage of end diastolic volume that is pumped out with each ventricular contraction. So at rest, most of you are at about 66, 67%, somewhere between 60 and 70%. And so what that means is that you're ejecting about 2/3 of the blood in your ventricle every time you heart beats. People that have cardiovascular diseases, their ejection fraction will fall. Okay? Their left ventricle is weak, it's going to fall. They're hypertensive, so pressure in the aorta is too high, it's going to fall. Okay? And so impaired ejection fraction is one of the ways that we can diagnose different kinds of cardiovascular diseases. Okay? During high intensity exercise, most of you guys can get to about 100% of ejection fraction. Okay? So there we go. All right. Now let's talk about cardiac output. Cardiac output is the product of heart rate and stroke volume. Right? So cardiac output is the amount of blood that is ejected or pumped out of the heart over the course of 1 minute. Okay? Stroke volume is how much leaves every beat. We know how many heartbeats we have in a minute. Multiply those two things together, and we get, okay, cardiac output. So very important to remember, cardiac output is over a minute. Stroke volume is per beat. Right? In an average person, cardiac output is about 5 to 6 liters of blood per minute. Okay? 5 to 6 liters of blood. Again, that's going to vary a little bit. If you're a smaller person, you have a smaller blood volume. Okay? If you're a very large person or a very highly trained person, you have a little bit larger blood volume. So imagine, right, Taylor has two 2-liter bottles of blood in her body. Think about it that way. Okay? Or maybe Zach's got 3. Okay? Actually not that much blood in your body. Think about it in that particular way. Right? You've got like 2 2-liter bottles, 3 2-liter bottles of blood. That's going to be. So cardiac output is normally 5 to 6 liters at rest. Your blood volume is normally going to be about 5 to 6 liters at rest. At rest, you circulate all the blood in your body through the heart about 1 time every minute. Okay? About 1 time every minute. During exercise, in an average person, it may reach 20 liters per minute. In very, very elite athletes, it may be 35 liters per minute. Okay? So we're going to increase heart rate. We're going to also increase stroke volume some during exercise. So that's going to make our cardiac output. The person that has the largest cardiac output, right? The larger your maximal cardiac output during exercise, the better an endurance athlete you're going to be. Okay? So, if all of you are roughly the same age, so we assume your age predicted max heart rate is the same, what cardiovascular parameter is going to separate my not so great endurance athletes in class from my better ones? Which one you want to guess? Stroke volume. Okay? Whichever of you has the largest stroke volume will probably win the race. Okay? You want to know how I can predict who has the biggest stroke volume? Whoever has the biggest heart. So I'll make the dad joke that in this way, in endurance athletes, the person, if you're all the same age, the person that literally has the biggest heart probably is going to win the race. Okay? So stroke volume becomes really important because in people that are all the same age, the heart rate component of cardiac output is the same. So whoever's got a bigger heart and a bigger stroke volume can have a larger cardiac output. Cardiac output pretty much determines VO2 max. And so there we go. Okay. We used to measure this in one of our labs. We don't do that anymore because you guys did it in some other ones, but we're going to talk a little bit about blood pressure. So blood moves by differences in pressure, so we need to understand kind of the pressure in different arteries and arterioles, as well as in your heart over time. Okay? That be a question? Mm-hmm. Sure, and I just mean, yeah, and endurance athletes can probably have, for their body size, going to have a larger heart than a non-endurance athlete as well. So, I guess I was sort of thinking that people that aren't endurance athletes don't really care what their VO2 max is, and therefore may not care if they can win a race or not. So it was not that there's some sort of physiological difference between those things. The idea holds across everybody. If you have a larger left ventricle, you'll have a bigger stroke volume. Therefore, you're likely to have a larger cardiac output than somebody that's the same age. Okay. What did I want to say about blood pressure? I've lost all train of thought. Somebody remind me. Somebody? How many of you have taken your blood pressure? Right? Blood pressure comes in 2 numbers, right? You get a larger number that we usually say first, that stands for systolic pressure. And then you get what is typically a smaller number second, which is called diastolic pressure. Systolic pressure is typically, when we measure blood pressure, usually in the brachial artery here, kind of under your bicep. Systolic pressure represents the pressure exerted on the walls of your brachial artery during ventricular contraction. Okay? Left ventricle contracts, that moves the blood, that's going to lead to an expansion or a raising of pressure in the brachial artery. You can measure that. Then during ventricular diastole, so the ventricles are not contracting, they're relaxed, pressure in those arteries is going to fall, right? That's going to be our diastolic. So we look at these two numbers to tell us. Okay? You can also calculate, although it's done much lesser now, something called mean arterial pressure, which is meant to denote the average pressure exerted by blood as it travels through our arteries. In a resting person, since you spend 2/3 of the time in diastole, only 1/3 of the time in systole, mean arterial pressure, you use this formula, but it basically takes systolic pressure and counts that for 1/3 and diastolic pressure and counts that for 2/3, and you get some average pressure exerted on those walls. Okay? Elevated systolic or diastolic blood pressure is what leads to hypertension, which is one of the most common forms of cardiovascular disease. Okay? If we took all of you in class, okay, I'm sorry, Mitchell, Zach, Cade, it's going to be one of you three or me. We're probably one of us, there's maybe like two more, one of us is probably at least like mildly hypertensive already. Men tend to have greater risk of hypertension than women. Usually when we get in class, we get like one guy that was kind of on the borderline of being kind of mildly hypertensive. Okay? So there are specific cutoff points for this that we will talk about. But what this means is when blood pressure goes up, okay, because blood moves via differences in pressure, it means that the heart has to work harder. It has to contract more forcefully to raise pressure in the ventricle to exceed pressure out in the arteries to be able to force the blood to move. Okay? As that happens, that can lead to some bad adaptations in the heart. As the pressure also rises, if you have weak points in arteries somewhere in your body or in your brain, it can lead to more strokes. It can lead to additional kind of things like peripheral arterial disease or heart attacks and those kinds of things from ruptures in coronary arteries. So having elevated blood pressure can be very dangerous, especially at rest. However, during exercise, okay, systolic pressure goes way up, and that's fine. It's perfectly healthy, it's perfectly normal, it's perfectly safe. Diastolic pressure ever goes up, we got big problems, we got to call 911. Okay. So in response to exercise, systolic pressure increases, diastolic stays right about the same. In exercise stays, Dr. Mike will explain to you guys the eyes. If we look at pressures, okay, the closer an artery is to the heart, the higher its pressure is going to tend to be during systole. So aortic pressure is a little bit higher than brachial pressure. Brachial pressure is a little bit higher than radial pressure as we move our way down. Okay? And so that's going to help us force blood away from the heart, out into the extremities, down into the capillaries, and then it's going to begin to collect back in the venules and the veins, and then return itself back into the right atrium. Okay? Questions about those? You doing okay? We'll talk about the blood for a couple of minutes, then we'll take a little break. Okay? And then we'll do a little bit of an activity. So I mentioned the other day that I think blood is maybe the most under discussed parameters, especially as it relates to exercise in the cardiovascular system. So blood is made up of these specialized cells and then what is basically water. Okay? And so plasma is the water or water-ish portion of your blood. Under normal kind of non-elevated altitude situations where you're not Lance Armstrong, you're not taking erythropoietin to make extra red blood cells, and you're lying about it, plasma is somewhere between about 55 and maybe 60% of your blood volume. Okay? So more than half of your blood is just water, or it's water with some ions. Okay? Levels can change acutely due to water loss and can also increase greatly due to exercise training. When you sweat, the fluid you are sweating out typically is going to be water coming out of the blood. Okay? It's water being forced out of those kinds of things. It's going to come from your sweat glands, but you're going to lose water out of the blood, and then it's going to move out through the sweat. Okay? Now, the mostly red blood cells that are going to be in your blood, that's hematocrit. Okay? Hematocrit technically denotes red cells, white cells, and platelets, but about 99% of hematocrit is red blood cells. We'll talk about the structure of red blood cells in a second. Okay? So we will sometimes talk about a parameter called your hematocrit percentage. So red blood cells are usually somewhere between maybe 35 and 50%, okay, of your blood volume. If you're anemic, right, which tends to be more prevalent in women, ladies, some of you are probably anemic or close to being anemic. You guys may be like under 40% on your red blood cells as a percentage, okay? If you are either very dehydrated, you've lost some of the water that's in here, if you live at altitude, or if you're a blood doper, okay, then your hematocrit percentage may be closer to, say, 50% as a percentage of total water. Okay? This matters, the hematocrit percentage, as well as the kind of total amount of blood that we have matters a great deal for determining exercise. Okay? And part of the reason that that is the case is because of red blood cell specialized structures that allow them to bind a tremendous amount of oxygen. Okay? So one of the big things that blood does is it lets us bind oxygen in the capillaries in the lungs and then circulate that oxygen out to the tissue so we can use it to make energy. Okay? And so the red blood cells have something called hemoglobin in them. And in a hemoglobin molecule, which has iron in it, it's able to bind 4 oxygen molecules. You have about 250 molecules, million molecules of hemoglobin per red blood cell. So a single red blood cell can bind 1 billion molecules of oxygen. You have hundreds of millions of red blood cells in your body. So you can get the idea of how much additional oxygen you can bind. Okay? So without hemoglobin, our O2 carrying capacity, or our ability to carry oxygen in the blood, would go down very, very dramatically. So imagine this particular scenario. I'm an endurance athlete. If I have more red blood cells, I can carry more oxygen in my blood. If I have more oxygen, I can take that oxygen to my muscles and turn it into energy. And therefore, I can bike faster or farther or run faster or farther. There's some really easy ways to make my red blood cell count go up. One, I just take a shot of a hormone called erythropoietin. It's normally made by the kidneys, and it makes your bone marrow make more red blood cells. Okay? Or I can go give blood. They can hold on to my blood. They can spin my blood down, separate the plasma and the red blood cells. Wait, 3 months, let my blood volume come back to normal, then they just give me back my red blood cells. Give me a blood transfusion. That will give me more red blood cells. If I have more red cells, I can carry more oxygen, I can be a better athlete. This is indeed what basically all of the professional cyclists that ride in the Tour de France do. Okay? Pretty much all of them do this. They take erythropoietin. You can't trace it. What they've done now is you have what's called a physiological passport. And so I would grab you on any random day, do a finger stick, figure out what your hematocrit percentage is, and then I would check that like during the off season and various things, and we build up over the course of a year sort of where your hematocrit percentage is. And if you deviate from that more than a couple of percentages up, then you can get disqualified. Also, naturally with physiology, you will never see a person over about 53% red blood cells in your blood. So if you're over that, you're clearly obese. This is how they're trying to combat that. If you want to get as much hematocrit in your blood as possible, go and live on Pikes Peak for a while. Go stay at altitude for a couple of weeks. And it will stimulate those kinds of things. Okay, let's pause here. We'll take a little break. Then we'll come back and we'll do a little activity, and we will continue on. As it relates to red blood cells, as it relates to hematocrit percentage, okay, what we're also really concerned about because of this is the way that oxygen is going to be carried in the blood. Right? Because 98% of the oxygen in your blood is carried on hemoglobin, and only about 2% is carried in just dissolved in the plasma. That's why having so many more red blood cells and therefore so much more hemoglobin can be potentially beneficial. Okay? So having hemoglobin lets us transport about 70 times more oxygen in plasma alone. Okay? And the other really interesting thing, I meant to bring it this morning, and my daughter was playing with it, and I forgot to bring it. So you guys have had what's called your pulse oxygen measure. You go to the doctor, put a little thing on your finger, it shines an infrared light in, and it tells you like you're 95% saturated, 98% saturated. Okay? So if you have normal pulmonary function, so you don't have asthma or COPD, and you've not gone to a very high altitude where there is less air and therefore less oxygen concentration, then typically hemoglobin in a normal person is going to be somewhere between like 95 and 98% saturated. And so one of the markers of if you have COVID and you're about to be sort of in crisis is that your O2 sat begins to fall precipitously. If you get into the low 90s, you get into the high 80s, then you need to go to the hospital immediately. That's why we have the pulse oxygen. We bought it last year in like April because they said that was a way to check if you were to get COVID and kind of check on things that's relatively cheap and expensive. Right? So the 98% saturation is way more than what we need. It's way more. Okay? And so we have this very high capacity if you have pulmonary function. That lack of oxygen is rarely going to be a problem for tissue functioning at sea level at rest. Okay? So, and then even if you are at altitude and even at very high intensity exercise, a lack of oxygen is rarely going to be the thing that really stops you and kind of limits performance. But what it does do, we don't talk about metabolism in class at all, by having more oxygen around, if you have more red blood cells and therefore more hemoglobin, is it allows you to make a greater percentage of energy for any given work rate from aerobic metabolism that doesn't make metabolic byproducts that are going to cause you to stop. So that's kind of the big issue. So conversely, also in your blood is going to be carbon dioxide. We use oxygen, we make energy, it also then makes carbon dioxide. And we have to get that carbon dioxide from our tissues back to the lungs so that we can breathe off that CO2 out into the air. Okay? And so we have some carbon dioxide that is just dissolved in the blood plasma, maybe up to about 10%. You guys may or may not know this, but you actually bind carbon dioxide onto hemoglobin. And so it's called carb amino hemoglobin, and that may be somewhere between 20 and 30% or thereabouts. And then the rest is going to circulate in your blood or in the plasma as something called bicarbonate. Right? When you mix CO2 with water, lots of water in your blood, CO2 gets in the water, it makes something called carbonic acid, right? Which is H2CO3. You put an acid in water. Just again, you make the acid, it's in plasma, the plasma is water. You mix the acids with water, they will dissociate, and we get hydrogen ions, which makes it acidic, and we get HCO minus, which is called bicarbonate. Okay? Anybody know what else has lots of bicarbonate in it? When you have an upset stomach because of excess stomach acid, what might you take? Tums. Very good. Tums is mostly just bicarbonate with some, fun like cherry flavor or something, right? And so you can drink Malox or Malanta or just pick something for upset stomach that often, very, very often has bicarbonate. So your blood, by carrying bicarbonate is a base, okay? Your blood can function as a buffer to soak up hydrogen ions from other parts of the body, okay? And so you can run this reaction in reverse. If you have excess hydrogen, it binds to the bicarbonate ions, and it drives the reaction back this way. You're going to get extra CO2 to be able to figure it out. So this is one of the ways that we can use blood and the cardiovascular and the respiratory system as sort of a buffer to maintain acid-base balance. Okay? So you guys have heard of something called lactic acid, yes? Lactic acid is something that is formed at the end of glycolysis, which is a type of anaerobic metabolism. You make lactic acid, make a little bit of energy. That lactic acid gets into the plasma or into your interstitial fluid, and it dissociates into lactate hydrogen. It makes things more acidic. Lactic acid can be buffered by bicarbonate. The hydrogen ions from lactic acid, that acidity, cause your muscles to burn. It causes you to want to stop exercising. So if you can soak up some of it like this. There's research that says if I give you a bunch of Tums before you go do high-intensity sprinting exercise that makes lots of lactic acid, I can raise the bicarbonate level in your blood, and you can buffer more lactic acid, and therefore you can sprint longer or faster in that way. Problem with that is that you're also sprinting to the bathroom because that much bicarbonate leads to diarrhea. But little consolation, right, if you win a 200-meter race or something at the Olympics. So this is kind of one of the things we can use physiology. Okay, so this is just showing a little bit of gas exchange at the tissue level. Here's that capillary blood. You've got the respiring tissue. This could be muscle. It could also be inside the lung. It doesn't really matter. I guess in this instance, this would be like muscle here. Here's a capillary running by that muscle. O2 unloads off of the hemoglobin in the red blood cell and goes into the muscle. CO2 that we're making in here is going to load onto the red blood cells and hemoglobin, right? We're going to load some in as well. We're going to dissolve some, and we're going to put some food in that carbon. Just kind of showing this process that is happening. Okay. Now let's talk about the vascular system. We talked about blood and kind of how we carry O2 and CO2. Let's talk about blood vessels. Right? Let's talk about blood vessels. Arteries carry blood away from the heart. Arteries are large blood vessels that carry blood away from the heart. Arteries are going to have some specialized structures on them. They're going to be surrounded by smooth muscle, which allows them to vasoconstrict or vasodilate, so they can get bigger or smaller. They're very stretchy. Okay. Arteries are going to, as they get smaller and smaller, they're going to branch down into what we call arterioles. Arterioles are going to branch and branch and branch and then form capillaries. Capillaries are very, very thick, like one cell layer thick. They have lots of holes or pores in them. They're called fenestrations, and that allows stuff to pass in oxygen, CO2, glucose, fat, you name it, we get. So we've got capillaries around an individual tissue. Capillaries come back together and make venules. Venules make veins. They bring blood back to the heart. Okay? Blood in arteries or arterial blood has high oxygen, low CO2. Okay? Blood on the venous side, so venules and veins, has lower oxygen, higher CO2. So that oxygen has left the blood at the capillaries, and that CO2 has entered the blood at the capillaries. So this is the actual side of what we call gas exchange. We use arteries, and especially arterioles, to direct blood to certain tissues over other tissues. Okay? Direct blood over certain tissues and send it to other places. Really, the only job of veins is to kind of store blood and send it back. So arterioles are what we call resistance vessels. And this is going to be, these are the vessels that are going to control blood pressure. And these are the vessels, by controlling blood pressure, that are going to control where all the blood goes in the body. Okay? We could pick someone. I'm trying to think. So we could ask Maddie to stand up on her desk and sing the national anthem for us. And the mask make it not work. You don't have to stand up. Okay? Saved by the mask. But we do this, right? If you've ever gotten flushed, right? Because it's a stressful situation or you're embarrassed or something and you get flushed, what is happening is arterioles that send blood to the skin are getting bigger. They're vasodilating. They're directing blood towards the skin. Okay? When you get cold, the arterioles that send blood to the skin will constrict and get smaller, and they will direct blood away from the skin, which is why sometimes when you get cold, you may appear less flushed. You may appear kind of pale and very white or something like that, if that is your particular skin tone. Right? And they do this because they have what's called vascular smooth muscle cells. Right? And this is a picture of vascular smooth muscle around the artery. So if blood vessels are tubes, okay, you have smooth muscle that goes all around the outside of those tubes that are your blood vessels. When that smooth muscle contracts, it squeezes the blood vessel down. Okay? How many of you have ever tried to shoot water out of a garden hose at someone? Okay? If I want to make the water shoot farther and faster, what do I do with my thumb or my finger over the end of the hose? Push down. Very good. Okay. When I close, I put my thumb over the end, and it makes the place where the water is leaving narrower. Okay. By narrowing it down, I raise the pressure, and then there's more pressure to make the water go far. Okay. That's the same idea of what happens inside arteries and arterioles. When you vasoconstrict and you squeeze down, the pressure goes up, which then means that blood pressure has to be higher in order to drive blood into that tissue. And if you don't get a corresponding change in, say, what's happening in the ventricle, then blood's just not going to go there. It's going to travel the path of least resistance. It's going to go to the areas of the lowest pressure. Conversely, when I want blood to go to a tissue, I relax the smooth muscle, and those arteries and arterioles get bigger. The pipe gets bigger, so the pressure inside of them falls. The pressure falls, then it's easier for the blood to move through there, so I'm going to get more blood through. And so we use this ability to constrict and dilate arteries and arterioles especially to help us decide where in your body are we going to send blood at any given moment in time. Okay? Right now, you guys are resting. Most of the blood in your body, or about 60% of the blood in your body, is going to your stomach, your intestines, your liver, and your kidneys. Okay? It's called the splenic region. When you eat and you get food into your stomach, more blood goes to your stomach. Okay? And then to your intestines. We're going to digest, and then we're going to try to absorb. When you exercise, and you start contracting skeletal muscle, immediately we vasodilate to send blood to the muscles, and we vasoconstrict to stomach, intestines, kidney, liver. Okay? And this is part of what we call a fight or flight response. And the idea is that traditionally, if I need to run or fight something off, it doesn't do me any good. I don't need to digest food if I'm about to get eaten by something, right? And so that's how we're going to control all this. We're really, really good during exercise, shunting blood away from all other tissues except for the heart and the brain, and everything else goes to skeletal muscle. And so we can send lots of blood and lots of oxygen to our muscles. This is just going to show you the arteriolar tree. We've got primary arterioles, we've got secondary ones, and they branch off at perpendicular angles. If we get all the way down here, these little bitty things which are going to be. Okay. Here is an electron micrograph showing pictures of vascular smooth muscle around an arteriole. Looks kind of like these little Michelin tire man kind of rolls that are going to be there. So these little rings of smooth muscle. The cool thing too about all of this is that in this one, they dissolved the arteriole and the muscle away, and all they left was this is nervous system innervation for this piece. And so we have sympathetic nervous system innervation that connects to the vascular smooth muscle. And what happens is when you release epinephrine or norepinephrine, it's going to tell the vascular smooth muscle to contract. One of the ways that we signal all of this stuff, it's going to tell it to contract. Okay? Anybody ever seen the show? It used to be on the Discovery Channel. I'm sure it's streaming somewhere else. It's called Mythbusters. Have you seen the show Mythbusters? Okay. I loved Mythbusters for a host of different reasons. One of the things that they did in one episode of Mythbusters, I think they did it because they wanted to scare people, was you guys have heard of the saying, right? The person got cold feet, right? What happens when you get scared and you don't want to do something, you back out of it, you got cold feet. They tested to see when you are scared, you actually get your feet drop in temperature. And so, of course, the conceit was then they put the people on the show like in a thing with scorpions and tarantulas, and they really tried to like, whatever it is that you're the most scared of, we're going to put you in there with those particular things. But they measured with an infrared camera, they measured the temperature and the blood flow going to a person's feet. And what they found was that when you get scared, your body releases epinephrine and norepinephrine, which causes vasoconstriction, and it causes vasoconstriction in the arterioles in your feet. And so you acutely see this drop in temperature because of a lack of blood flow going to the feet. And so I thought it was cool because one, it was some fun cardiovascular physiology, but also, people had like snakes and tarantulas and all of which just absolutely just terrifying. So that's kind of what's going to happen. Again, in exercise physiology class with Dr. Mike, we'll talk a lot more about how exactly exercise changes, and it's going to cause stuff to not go to the splenic regions and cause it to go to skeletal muscle. And that's preserved in the brain, and that's preserved in the heart. Okay? Capillaries and veins, those are pretty self-explanatory, but they're not nearly as fun. We talked about this a little bit. Blood mostly at the liver and the kidneys right now. You're cleaning and processing the blood in the liver, and you're making urine. Kidneys, talk about what happens during exercise. Those things are pretty straightforward. This is just kind of a cartoon showing all of that. Okay. Nope. Okay. I'm going to mention this parameter. Okay? I'm going to mention this. But then we're not going to come back to it and really make much use of it until we get into talking about aerobic metabolism and aerobic metabolic determination of exercise performance, which will be the next things that we do. So I'm going to mention this. It may kind of as a definition show up on the test, which is really going to become much more important when we get into talking about things that determine performance. It's a parameter that's called arterial venous oxygen difference. Okay? It is very often abbreviated as AVO2 diff. All that this tells us is how much oxygen has left arterial blood at the capillaries and gone into a particular tissue. Okay? It is a way for us to measure something called oxygen extraction. How much oxygen has left arterial blood in the capillaries and gone into the tissue. So you know how much oxygen is in arterial blood, it goes to a tissue. You know how much is in the venous blood leaving that tissue. The difference in oxygen just tells us then how much that tissue took up. Okay? It's a very important parameter, but it's also one that is fantastically difficult for us to measure. Okay? Fantastically difficult for us to measure. I can measure hemoglobin saturation, no problem. I can measure heart rate, no problem. Okay, we can do that right now, non-invasively here in class in 2 minutes. If I have an ultrasound machine, I can do an echocardiogram and I can measure stroke volume. I can measure left ventricle size using the ultrasound on the heart. A little bit more trouble, but not that big of a deal. If I want to get at AVO2 difference, I have to run a catheter into an artery and a vein leading to and going away from the same tissue. Okay? So if I want to calculate, let's just say, the AVO2 difference in your quadriceps muscles during cycling or running, I have to run a catheter into your femoral artery and a catheter into your femoral vein. Then I have to take a sample of blood from the femoral artery and a sample of blood from the femoral vein. And that would let me calculate then roughly how much oxygen your quadriceps are taking out of arterial blood and using during exercise. Okay? Any of you guys ever had a venous catheter run? Then you surely haven't had an arterial catheter run. Okay? I could run, if you guys would let me, I could run a very simple venous catheter. It's like you go to give blood and they just stick you and then you insert a little Teflon catheter in and it has a little stop top on it and you can open and close it and you can sample venous blood. It's not that big of a deal in a vein because the pressure is so low. If you're a trained phlebotomist, you can put venous catheters in just like drawing blood. It's not a big deal. Arterial catheters, because the pressure is so high, generally have to be set by a cardiology nurse or a cardiologist. So you have to find, like, say, my brachial artery, and I have to puncture the artery with a catheter to be able to get it in there. And the problem is, if you miss or you can't get the catheter in, then blood just like spurts and shoots. We had a cardiologist when I was a master's student, one of the PhD students in our lab. They were running an arterial catheter. They were going to do an infusion of a particular drug that would lead to vasodilation and vasoconstriction. They had a cardiologist there to do it. The cardiologist missed getting the catheter set, and our participant was like bleeding all over the lab, like right there. We had to shut the project down and get the cardiologist to move. It was crazy. The key is that the cardiologists don't actually do the lines very often. It's always the nurses that do them for the most part. The nurses are the real heroes in this whole thing. Cardiologist kind of comes in and does the surgery and monitors things, but that's how we're going to do that, okay? It's a really important parameter because if I know your AVO2 difference, I can estimate things about aerobic metabolism directly occurring in skeleton muscle. I can tell you things about how many capillaries you may have. I can tell you things about how well the mitochondria in your muscles function, but it's almost impossible to measure. You don't typically do it during studies here in the US. So you go to Scandinavia, and they're just like, whatever, fine. You just let anybody run an arterial and venous line. So they do all kinds of crazy things. Their willingness to allow stuff for research purposes is a little more relaxed. Okay? So we'll talk about this in the context of cardiac output and how we can use cardiac output and AVO2 difference to predict VO2 max. To predict changes in VO2 max that may occur. We'll talk more about it. I just wanted to introduce it right now. Okay. Real quick sort of run through the respiratory system. Some books call it the respiratory system, others call it the pulmonary system. It's basically your lungs. Okay? It's basically your lungs. And so what I want you to know is very, very simple. We're not going to talk about bronchioles. We're not going to talk about the conducting zone and the ventilating zone and the respiratory zone and all these other different kind of things that are going to denote where gas exchange takes place or it doesn't. We're not going to talk about the purpose of the trachea and the pharynx and the larynx and all of these kinds of things. Okay? I want you to know lungs, alveoli, and then the pulmonary capillaries that surround the alveoli. That's it. Okay? So in your lungs, there are thousands of these little individual air sacs that are called alveoli. They add surface area. That's what makes them so important. Okay? You have enough like lung surface area, it's like half a tennis court. Okay? Imagine that all like wrapped up in just right here. And that's because of these little alveoli. The alveoli are very, very vascular. They have a huge number of capillaries that surround each little individual air sac. So when you breathe air in, the alveoli fill up with air, right? And then there's going to be more oxygen in the air than there is in this pulmonary capillary. So oxygen will diffuse out of the alveoli into or onto the hemoglobin on the red blood cells in those capillaries. And then that's going to move back to the heart and get sent out of the systemic circulation. Conversely, there's more CO2 in the pulmonary capillaries than there is in the air that you breathe in. So the CO2 will leave the blood and get into the alveoli, and when you exhale, when you expire and you push air out, then CO2 is going to be. Right? This is going to talk about mechanisms of breathing. Air, like blood, air moves via differences in pressure. And so when you want to breathe in, right, you're going to contract your diaphragm. You contract your diaphragm, it lowers itself down. So big, deep breath in. Diaphragm falls. The area inside your chest cavity gets bigger. That means the pressure drops. And then you open your mouth or your nose. And so air is going to go from higher pressure outside to lower pressure in. So it's going to move in. So you're going to bring room air into your lungs. When you want to exhale, then you're going to relax your diaphragm. It goes back up, squeezes that space down, pressure raises. Pressure inside is higher than pressure outside. So then air is going to. Typically at rest, breathing is relatively passive. We'll talk about the respiratory centers and what controls diaphragmatic contraction in a second. But at rest, you just get this natural, not thinking about it, diaphragm goes contract, relax, contract, relax. Okay? If asked to do so, you have some level of voluntary conscious control of your diaphragm and your internal and external intercostal muscles. And so you can forcibly bring more air in or expel more air out. Okay? But again, all that does, when I forcibly breathe in, I'm going to open the chest cavity up even more and drop the pressure. When I forcibly exhale, I'm going to squeeze it down even more than normal, raising the pressure inside the stomach. Talk about those. So, why is it that in some situations breathing goes up and in others it goes down? We're going to do a little experiment. Okay? A little experiment. Everybody put whatever it is that you're doing down. I'm going to put my stopwatch out. Okay? We are going to hold our breath for 30 seconds if you can. Now, admittedly, you have a mask on, right? And you take your, we're going to take the word, you do whatever I want you to do. Get ready? We're going to hold our breath for 30 seconds, and I want you to then pay attention to what happens. When I tell you to stop, watch and pay attention to what happens to your breathing right in your gut. All right? You guys ready? All right. 3, 2, 1, go. Stop. Okay. Let's try. Yes. Well, what happens to your breathing right when you stop? Okay. Took a big deep breath in. Okay. Good. Anybody else have a different thing that happened right after all of that? Samantha, what about you? Anything else? One big deep breath. 2, 3, what happened? Okay, well, when you had to finally let go and breathe, what was that like? What happened then? Okay, like just normal breathing. Interesting. Okay, so what's happening while you guys are holding your breath that's making you feel that urge to want to breathe? Anybody know? CO2 is building up. CO2 in the breath. Okay? The primary regulator of breathing is CO2 concentration in arterial blood, which is going to seem weird, right? Because you think it would be CO2 in venous blood, it's actually CO2 in arterial blood. When you don't breathe out CO2, its levels in arterial blood rise. That interacts with receptors in your carotid arteries, okay, and in the brain, and it signals to what are called the respiratory centers in the brain, which are going to urge you and make you want to breathe. And therefore, you have to consciously counteract that so that you don't breathe. Okay? So, the regulation of breathing is mostly controlled by areas in the hypothalamus that are called the respiratory center. So you have motor neurons that are going to go to the diaphragm, right? The respiratory centers are going to send out periodic signals, much like the SA does. So you normally at rest don't have to think about breathing, okay? The respiratory centers send out firings to these motor neurons, diaphragm contracts and relaxes a few times a minute, no matter what. When we sense chemical changes like rising CO2 or rising hydrogen, okay, it signals an increase in the rate and the depth of breathing. Right? So you're breathing becomes deeper, big deep breath, and then oftentimes its rate is going to go up. This is going to cause the respiratory centers to change their signals by what we call chemoreceptors. Okay? Things that are going to sense increases in CO2 and increases in hydrogen. They tend to be in the aortic arch and the carotids, and they're going to signal these things. Additionally, you have stretch receptors in the alveoli and in the bronchioles that if you take too big and too deep of a breath, you're going to stretch these things too much, and that's going to then lead to activation of these receptors, which is the opposite. They shorten the duration of breathing and make the breathing not quite as deep, but they need to get stretch. So, during exercise, your breathing rate and depth goes up. Your ventilation level goes up. Okay? It goes up because you're making, for the most part, you're making more carbon dioxide and skeletal muscle in the metabolism. You know, for arterial CO2 is going to go up a little bit, and that drives an increase. Okay? Same thing happens, it's just like holding your breath. Okay, we talked about this, we talked about ventilation, blah, 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 phase 1. So the brain is very, very sensitive. It's very sensitive to changes in CO2. Also has a little bit of a sense of movement. You guys have things in your joints called proprioceptors that sense position or changes in position. You activate proprioceptors, it also leads to an activation of the respiratory system. So when you start moving your arm, you're going to get an immediate kind of a little bit of an increase in breathing. It's going to take place because of that. So phase 1 ventilation increases are very quick. No changes in the chemical environment. No bodily movement. In phase 2 is more gradual, and that's going to be different. I don't really want to talk about these kinds of things. Blah, blah, blah. Oh. You know what? We're going to stop there. So we'll pick up on the cardiovascular responses to exercise on Wednesday. It'll take us 5 minutes. Basically, everything changes in the stuff. That's it. So we'll pick up there on Wednesday, and then we'll start talking about aerobic training. Right? Remember, if you owe me data for lab, I've gotten an email from one person. You owe me data from lab. I need it today. Come talk to me about it. Send me an email. Send me a text message. Okay. Lab is due Wednesday. Resistance training fatigue is due tonight or midnight. All right. Very good. We'll see you guys on Wednesday.